Started. Good evening on this lovely spring evening and day here in New England as we are preparing for opening day tomorrow. I hope you're all paying attention to that. This has been a crazy spring, hasn't it? Anyway, my name is Jim Wessler, and I'm the CEO for the Massachusetts, New Hampshire chapter, and I'm really thrilled to welcome you to our annual Spring Research Forum. This is really a terrifically well-received, and I think hopefully you'll agree when you hear all of our wonderful speakers. A great evening. Before I begin, I would like to acknowledge we have a lot of wonderful staff, but Kelsey Goslin, who is our manager of medical research engagement, was probably the primary person that was involved in organizing tonight's event. And Kelsey was somewhere. There she is. Kelsey, you want to raise your hand? So we have a really pretty simple and straightforward vision at the Alzheimer's Association, and it's a world without Alzheimer's. And we will achieve that in part through aggressively implementing our global research program. You probably know, or many of you know, the Alzheimer's Association is the world's largest private nonprofit funder, the world's largest of dementia-related research. Since our inception, the Alzheimer's Association's International Research Grants Program has awarded more than $405 million to over 2,600 scientists worldwide. We fund globally. And here in Massachusetts, we are currently funding 35 grants across 11 institutions totaling approximately $9.5 million. That's in play right now here in Massachusetts. So in addition to funding cutting-edge research, the Alzheimer's Association is also expanding our work, lost my train of thought here we go, to actually funding our own research studies. So currently the Alzheimer's Association is advancing science with 3 large-scale studies that I want to mention to you. Due to achievements made in developing biomarkers that enable doctors to better identify the disease, we now have a technique called amyloid PET imaging. I imagine you'll hear a little bit about that in a little bit. The Alzheimer's Association is helping lead a research initiative called the IDEAS Study to determine the impact of whether receiving this brain scan improves a patient's care plan, medical outcomes, with the ultimate goal of getting those scans covered by insurance, which we certainly hope Medicare will cover that eventually. The Alzheimer's Association is also funding and leading the longitudinal, these are great names, the Longitudinal Early Onset Disease Study, or LEADS, with the goal of enrolling a large cohort of individuals with early onset Alzheimer's disease in order to provide longitudinal clinical and biomarker data to ultimately improve care detection and intervention. Now, one of the study sites, which we're pleased to share with you, is here in Boston, and it's being led by none other than our Medical and Scientific Advisory Co-Chair, Dr. Brad Dickerson from Mass General Hospital. Now, finally, there is an accumulation of research showing, and you're going to hear more about this as well this evening, how lifestyle interventions like diet and exercise and social stimulation may be able to reduce the risk of cognitive decline. So the Alzheimer's Association has just launched, we're literally in the last few months, I think, Cynthia, is that right? We're just launching this thing, called the US Pointer Study, with a $20 million commitment that we have made at the Alzheimer's Association. US Pointer is a 2-year lifestyle intervention trial to support brain health and prevent cognitive decline in older individuals at increased risk of dementia. What is really exciting about this study is that the intervention will be delivered at the community level through local Alzheimer's Association chapters, all the study sites have not been announced yet, and local YMCAs, which is particularly, I think, important because this could lay the groundwork for what might be then a much larger rollout if the intervention proves to be successful. So in addition to funding and leading research, the Alzheimer's Association also provides premier global forums for the greatest minds in Alzheimer's and dementia science to collaborate, connect across disciplines, address common challenges, and share new discoveries. We, the Alzheimer's Association, host the largest dementia conference in the world, bringing together over 5,000 of the world's top dementia researchers to share these ideas and present their work. Last summer it was in London, and this summer the conference will be in Chicago. But while we diligently work towards finding effective treatments, we are also equally committed to the goal that we must do everything possible to assure that every single American currently living with dementia and those to come have access to the best quality care and support. We have a range of services, and if you're not, I know some of you have been coming to this for years. If you're new to the Alzheimer's Association, we have information table that our staff will be there at the end of tonight's program to get more information. We have a range of services and programs that help families who are living with this disease. Our central entry point is our 24-hour helpline, and our 800 number is on everything that all of our literature. This is literally staffed 24/7. I challenge some of you to go home tonight after the program and you call that 800 number, and it's going to be answered. Alzheimer's Association, how may I help you? Family members, people living with dementia, and professional caregivers can call and receive the information and support that they need. We also deliver hundreds of educational programs, and we provide access to a large network of support groups throughout our 2 states and indeed throughout the nation. We, the Alzheimer's Association, have extensive programs for people living with dementia, and all of our programs for families are delivered at no charge to them. This disease has enough challenges as it is that ability to pay will not be a barrier from the Alzheimer's Association. We are completely a nonpartisan organization, but we are very active in state and federal affairs, as our elected leadership can have such an impact on this disease, on the progression, on research, and on care and support. Because of our successful federal advocacy, we have seen dramatic increases in funding to the National Institute of Health specifically for Alzheimer's and dementia research. In fact, under 2 weeks ago, the federal budget was approved, yes, it was approved, and signed into law, and because of our advocacy efforts, your advocacy efforts, we were successful as the National Institutes of Health were allocated an additional $414 million specifically for Alzheimer's and dementia research. This brings the annual NIH allocation for Alzheimer's and dementia research now to $1.8 billion, and that's because of your work. So this supports not only the scientists that you'll hear tonight, but we hope scientists not only locally, but certainly across America in their cutting edge work. We are doing everything possible to get the necessary resources so these researchers and scientists have what they need to achieve our vision, a world without Alzheimer's disease. Now, in both New Hampshire and Massachusetts, we have also been active on the state level. We have enacted laws and regulations that create quality of care standards that are improving the lives of tens of thousands of families in our 2 states. As but one example, because of our collective work, your work, over the next 3 years, all Massachusetts hospitals will need to become dementia capable. And currently, we have, and I don't have time to go into details on this, but I want to mention that we have an Alzheimer's omnibus bill that includes mandatory training for clinicians. It actually encapsulates what I just said about hospitals and state law. It's a bill that has many, many important pieces. It has the number H4116. We have information again out in the lobby. Please stop by the association. It is now in the state Senate. If you know your state senator, we need your help. We are literally moving the nation to support our cause, and we need your help. Whether you are a public policy advocate or a volunteer delivering our education and outreach programs, or whether you participate in the Walk to End Alzheimer's, or the Ride to End Alzheimer's, or the Longest Day, all of these efforts are vital to advancing our mission. And if you're just sitting here in the audience tonight and saying to yourself, gee whiz, I know you're all saying gee whiz, I'm not doing any of these things, well, it's never too late. We need you. Again, stop by our table at the end of the program, and you can learn how you can help. In fact, I've been moved, I'm sure many of you had, that over the past month, teenagers across our nation have demonstrated that democracy is not a spectator sport. And in a similar fashion, a lesson can be drawn for our cause. We cannot beat this disease if you sit on the sidelines. Please, please join us. So, thank you. So now it's to the real reason you're here tonight. I'm going to introduce our moderator, who's been doing this for really a number of years, I keep telling Brad he has another career as a talk show host or 60 Minutes or something like that, but he does a great job. But I want to introduce to you Dr. Brad Dickerson, who, as I just mentioned, has been a longtime supporter of the Alzheimer's Association and our local chapter. He currently serves as a co-chair of our chapter medical and scientific advisory committee. Dr. Dickerson is a behavioral neurologist and neuroscientist dedicated to the sophisticated, compassionate, and multidisciplinary care of patients with neurodegenerative disorders, specifically Alzheimer's disease. He is director of the Frontal Temporal Disorders Unit and the Laboratory of Neuroimaging at Mass General Hospital. In addition to teaching neurology at Harvard Medical School, he leads an annual course on cognitive neurology at the American Academy of Neurology, and he co-directs the annual Harvard Dementia course, which attracts primary care physicians really from across the country. He has received 2 research grants, I'm proud to say, from the Alzheimer's Association. The first in 2007 for a pilot study of ultra-high resolution MRIs and mild cognitive impairment. And the second in 2009 for his study of neuroanatomic biomarkers for dementia diagnosis. And I just learned this evening, about 45 minutes ago, that he's also a drummer and in a rock and roll band, so maybe he'll tell you about that. Brad Dickerson is a leader in the field of Alzheimer's research, and the Alzheimer's Association is so grateful for his passion and commitment to our collective cause. Dr. Brad Dickerson.
Thanks, Jim. You're giving away all my secrets. No, I'd really like to thank Kelsey Goslin for her tremendous efforts to put this program together tonight, and to Jim for his leadership in many national initiatives that he only touched on, and local initiatives as well. So we have an amazing chapter here, and I think many of you know that, but we'll touch on some of their roles tonight as well. So I wanted to just start by mentioning a couple of men that I've had conversations with in the last week. Fred is a 76-year-old man who was a teacher, and I met him and his wife 12 years ago when I diagnosed her with a posterior cortical atrophy variant of Alzheimer's disease, which affects vision rather than memory. And unfortunately, she passed away recently, and I was talking with him because he selflessly donated her brain to neuroscience. And so as a teacher, he felt not only are we going to learn more from studying what was going on in her brain, but we'll hear from some of our panel tonight about how studies in the laboratory of brain tissue from people and from animals and in artificial experiments, but can really give us insights into what's going on in the brain of people living with this illness. Earlier this week, I met Dave, who is a 62-year-old former engineer, and we just diagnosed him with a form of Alzheimer's disease at age 62. He's still very independent. He would easily blend into this audience, and you would never know that he has that diagnosis unless he told you. He's becoming an advocate. I think many of you know people like this, and I think we don't know what his journey is going to be like, but he and his wife are committed to joining research efforts, and we're working on seeing if we can get him into a clinical drug trial. So I think it's through the engagement of people like this and many of you that we're really able to make the kind of progress that we are and that we'll be trying to help communicate to you tonight, which I think we all feel a lot of hope from. And the point that Jim made about the increase in funding, it's amazing that we're now at a point where we're talking in billions rather than in millions, where we were even just 4 years ago. But it makes a real difference, and the place where I think it makes the biggest difference is in the careers of newly minted clinicians and scientists in the field, because it's traditionally been a difficult field to stick with, to stay in. If you're interested in it, it's hard to get that first grant. It's hard to sustain your faculty efforts as you come onto the faculty of one of the hospitals here or around the country. And I think it's the Alzheimer's Association that has really invested in that next generation of clinicians and scientists, and through some of those efforts, the NIH is now able to support more of those individuals as they embark on their own independent research careers that will keep them in the field. So I think it really makes a practical difference in what people spend their careers and their time every week working on. So it's really, not something that we should think about in the abstract actuarial sense, but something that really makes a difference in the lives of people that want to dedicate their professional lives to working in this area. So I want to just move now into a brief overview of some of the, let me just get the slide clicker out here. By the way, I've been saying to Jim, I think, and Kelsey the last few years, that maybe we need to either change the name of this or move it later in the year, because is it really spring right now? I think there's snow forecast for tomorrow. So maybe we should think about that. But we all maintain our hope about the coming of spring. And I like to start with a little art. And this painting was painted by a patient of mine who has frontotemporal dementia. He's been an artist for many years, but he continues to enjoy spending his time this way, even though his interest in it and some of the ways he does it have changed. But I think that the care consultation service and the coordination efforts that the Alzheimer's Association never has time to spend time in these forums talking about really can help people maximize their efforts to maintain quality of life through things like art, music, and other programs of that sort. So I'm delighted that we have on the program tonight Dr. Cynthia Lemire, Dr. Stephen Arnold, and Dr. Andrew Budson, who are going to help us understand a lot about a variety of different ways that we've made progress in this field and are making progress going forward. And to give just a little bit of foundation for some of what we're going to talk about tonight, I wanted to just touch on a few of the things that we'll, I think, we'll refer to over the evening. I don't need to say much about the care and support services, except to say that Jill Hovanazian and others in the Dementia Care Consultation Service are here, and we work very closely with them in helping to not only provide the care and support that we're doing now, but increasingly do rigorous studies of whether different forms of care that are being delivered are optimal for everyone, or whether there are some forms that are optimal for some people and some that are optimal for other people. So there's a fair bit of research now that is being funded with these new dollars that is not only focused on the science of the biology of the disease and imaging and clinical trials, but also on studies of the efficacy, the effectiveness, and the value of the programs and services that we talk about every day as making a difference in the lives of people with this illness. So in thinking about Alzheimer's disease and related disorders, we're increasingly starting to separate the pathology of the disease that is happening in the brain from the symptoms that people experience in their daily lives. And this is meant to capture the idea that when we think about Alzheimer's disease, traditionally we've talked about people that have memory impairments or other impairments in cognitive function that might be called mild cognitive impairment if they're still functioning pretty independently, or might be called dementia if the person has lost independence in their occupation or social functioning. But what we realize is that there's probably at least a 15 or more year time period while the disease is taking hold in the brain in what we call the preclinical stage, before there are any symptoms that are evident in the person's life, or maybe even on detailed testing that we could give them. And we'll be talking more about that because I think many of us see that as a window of opportunity to try new therapies, as is already being done. And the biomarkers of the disease that we'll hear more about tonight are measurements that can be made from living people that are thought to reflect so-called biological markers or biological processes going on in the brain. And the two big ones that we often talk about, and we'll hear about other ones tonight as well, are the amyloid plaques, which are on the top, and the tau-related neurofibrillary tangles, which are on the bottom. And these are things that you can see in the microscope, under the microscope, if you look at brain tissue, and that to some degree have been recapitulated in laboratory animal or cell models of the disease. But through spinal fluid, or increasingly through different kinds of PET imaging, we can measure some of these things in the living human brain and see where they are in the brain and see how much of them are in the brain. And that, I think, has really revolutionized a lot of the science that we're doing with the studies that you'll hear more about tonight. But the bottom line is that there's a very complex cascade of processes that we'll talk about a little bit shortly that relate the amyloid plaques and the tau tangles to what's referred to as the neurodegenerative process that involves much more than just those players when it comes to the molecules that are leading to the degeneration and the death of neurons or maybe of other kinds of cells in the brain as well. So this is a complex process that has many facets that we don't yet know how to measure in living people, but that we'll hear more about tonight from all of our speakers. So how do we move from our understanding of the disease in the laboratory full circle from the patient to ultimately testing new treatments? I think first we have to start with better scales to measure the symptoms. We often do what's viewed by many of us as fairly crude neurological exams or cognitive testing, and I think increasingly we're starting to move toward computer-based or tablet-based or other kinds of technology that will help us measure symptoms in a more granular way, in a more detailed way in people's daily lives or when we try to subject them to different kinds of tests. So that's a rapidly evolving science as we speak, taking advantage of a lot of the technology that you may have used to get here tonight or that you have in your pockets with you. We're increasingly able to measure changes in brain structure and function, the circuits of the brain that represent the firing patterns and the communication pathways that enable me to stand up here and think and communicate to you, and you to sit there and listen, see what's on the screen, and understand what I'm saying to you. These are circuits in the brain that enable us to remember things, to communicate with each other, to make decisions, to have states of mind like mood states and so forth. And increasingly, we're recognizing that some of these diseases, or forms of Alzheimer's disease especially, seem to attack certain networks. In general, you can look at networks in the brain with things like functional MRI scans, where any of us could get into a scanner and do a task, and we'd see different parts of the brain that light up, and those are associated with the function that we're doing in normal people. And if you look at damage to the parts of the brain that you can see in some people with forms of these diseases, which may be a form of Alzheimer's disease called progressive aphasia, the damage is really very similar to the network that serves that function. So at the beginning of some of the forms of these diseases, they may conform to the memory network, they may conform to the language network, or they may conform to one of the other networks in the brain that helps us do the things that I'm talking about. And the value of that potentially is that we can understand what's going on in a person's brain better and maybe be able to measure whether we're able to improve the functioning of the brain no matter what the exact kind of damage is happening to it. So that's one way that we're using some of these advanced brain imaging techniques. We're also doing what I mentioned earlier, which is increasingly being able to measure markers of the molecules that are the culprits of the problem in the brain. And the thing that we hear the most about these days that we'll talk more about tonight, but that is just the tip of the iceberg in terms of what we're beginning to be able to measure and use in the context of trying new treatments, is amyloid PET imaging. This was really the first key player in our ability to measure the molecules in the brain that are associated with the damage that's occurring specifically to Alzheimer's disease. And this was an amazing cover image on the journal Nature that came out a little over a year ago that showed an example of the amyloid that was present in the brain of a person when they entered this particular antibody trial. And then after about a year, there was essentially no measurable amyloid, at least in the brain of this individual and other representative individuals of that group. And so I think it really offers a clear and concrete example of how we can use these tools to move into therapeutic trials that tell us that we're hitting the thing that we think we're aiming for, we're hitting the target that we're aiming for, and we're changing it in a way that we hope will be helpful to the person. And of course, we then need to link that information to how the person is doing, and that's part of the subject of the larger scale version of this study of the so-called aducanumab antibody trial. That's one of a number of trials that are going on now that we'll allude to tonight. So then, how about laboratory models to understand disease mechanisms? Well, we may make laboratory models from animals, transgenic mice being one resource, or from cells that are grown in the lab, or from samples that you can obtain from an individual of skin and reprogram the cells to be so-called stem cells and turn them into a model in the laboratory of neurons, the brain cells or other kinds of brain cells that you can then do experiments with that are ultimately a human-derived model of the disease. And so this is a fairly new technology that we heard a little bit about from Rudy Tanzi at last year's Spring Research Forum, and a number of people in the Boston area are pioneers in doing this kind of work, including Dr. Lemire and others associated with her. And I think it's really another kind of technology that helps us model the disease in different ways in the laboratory than we used to be able to. And we can then use these laboratory models to screen drug treatments. And so if we've got measures of a protein like tau that are lighting up in yellow here in these neurons that are being looked at under the microscope, and we give a certain treatment, we can see if it reduces the amount of tau in that cell model of the brain. And that might be a lead for a new treatment that we could take further into tests that might tell us whether it's safe to give to people, and then whether it might be something we want to try to give to people in the next generation of clinical trials. And ultimately, then, we want to bring this back to people, and we'll hear from Dr. Arnold tonight about clinical trials of potential treatments, and from all of our speakers, actually, about different ways to try interventions that may not strictly involve drugs, but that may involve other kinds of interventions, including things that Jim alluded to in the Pointer study that we'll talk about with all of our speakers, at least touch on tonight. So that's really the life cycle of how we go from the process of assessing people to the process of learning more about the disease to the process of trying new treatments. This is a map of the NIA, the National Institute on Aging, funded Alzheimer's centers. There are many other places that do this kind of research around the country in university laboratories and in pharmaceutical company laboratories and elsewhere. But this is the network of almost 30 centers that have been really plugged together for now several decades to study this work and work in a collaborative way to advance our efforts within this country and also in collaborations around the world. And Jim mentioned the LEAD study, which I'm one of the principal investigators on, and this is an effort really to bring our studies into the people who are younger, who have sporadic Alzheimer's disease. They don't have one of the inherited forms. They seem to just have gotten it for reasons we don't understand yet. And these are people between the ages of 40 and 65 that will be enrolled in the largest study to try to understand more about what the disease, what happens with the disease in these younger people, many of whom are very interested in getting engaged in trials and may have fewer other illnesses that might complicate our ability to give people medicines or to judge whether they're working on the thing that we're aiming for. So the thing that I'm really excited about is that Maria Carrillo, who's the Chief Scientific Officer of the Alzheimer's Association and with whom I was a trainee in graduate school. When I was in med school, she was in graduate school at Rush University in Chicago. She's a co-principal investigator. She's really one of the scientists helping us lead this study, no pun intended, and is really bringing the full force of the Alzheimer's Association together with us to do this next generation study that I hope will help get more of these people on the younger end of the spectrum into trials. Many people in this age group have been excluded from trials in the past or may have atypical presentations and so haven't been able to get into many of the drug studies that have tried these new drugs. So, and I just want to conclude my opening remarks by saying that I think partnerships are the key. That's why you're here. Many of you are involved in different facets of partnerships through the Alzheimer's Association that ultimately catalyzes all of us to work toward the world without Alzheimer's disease, which is where we hope we'll be in the not too distant future. So thank you for listening to my opening remarks, and I look forward to bringing up our speakers. That's wishful thinking. And by the way, before I introduce our first speaker, I just want to mention that we're going to have a Q&A. So we'll have 3 speakers that I'll interview myself, and then we're going to have a question and answer panel with all 3 speakers up here. And Cards will be being passed around, as is being done now, pieces of paper that you can use to write down questions and pass them up here, and I'll do the moderating to select questions from the audience to ask our speakers after everyone is finished. So the first speaker tonight is Dr. Cynthia Lemire, who's a scientist in the Ann Romney, I think Cindy was going first, right? Yeah. Did I go past it? Yeah, sorry. Kelsey told me, don't forget to click forward to get the speaker's name up there, and I got ahead of myself. So Cindy is a scientist in the Ann Romney Center for Neurological Diseases at Brigham and Women's Hospital and an associate professor of neurology at Harvard Medical School in Boston. She focuses her research on translational research for understanding, preventing, and treating Alzheimer's disease, and as we heard, is a member of the National Medical and Scientific Advisory Council. Please join me in welcoming Dr. Lemire. Okay, so please feel free to shout out if there's any moment when we're not very audible. So just let us know. We want to make sure you can hear. So please start by maybe telling us a little bit about what got you interested in coming into the field of Alzheimer's disease and the research that you do. Can you just give us a little bit of the backstory on that?
Sure. My background was actually in teaching and teaching special education. Okay, let me see if I can tell you what I'm going to sit there.
You're not shy.
I know, that's good. Is this better? Is this better? Okay. So in the 70s and the early 80s, when I went to college, I was interested in special education, and I actually ended up teaching several different groups of Down syndrome kids. And I really loved that. And as I finished college, I became more interested in pursuing neuroscience and trying to figure out how the brain works. And eventually, I became interested in Alzheimer's disease research. I worked with one of the forefathers of Alzheimer's research, Bob Terry, in San Diego at UCSD. Then I moved to Boston, and I started working with Dennis Selko. And so I started graduate school and started working on looking at Alzheimer's disease progression in people with Down syndrome. So it was kind of coming full cycle. And I still continue to this day to investigate that process. In the 80s and the early 1990s, when I was doing my work in Down syndrome brain tissue, the average lifespan was around 30 years of age. And now it's around 60 to 65 years of age for people with Down syndrome. And with Down syndrome, it's called trisomy 21 because people with Down syndrome have 3 copies instead of 2 of chromosome 21. And the amyloid gene that Brad showed earlier, the gene that encodes that amyloid protein is on chromosome 21. So people with Down syndrome develop Alzheimer-like pathology quite early, and typically by the time they're in their 40s. And so because people are living longer, we see this as similar to some of your early onset studies where we're trying to understand better. If everybody has the pathology, everybody with Down syndrome develops the pathology in their 40s, why is it that some don't show signs of dementia until they're in their 50s or 60s while others show it in their 40s? And we think that this is a very important age and study to understand better what are the driving forces. Not everybody with plaques in their brain shows dementia at the same time. So we're trying to really understand what are the other processes that are driving the brain towards the dementia state.
It's amazing. I didn't know that you had taught people with Down syndrome before you got into this. It's amazing the way these things get connected. Can you tell us a little bit, you mentioned the amyloid as the key factor in Down syndrome that leads you to pretty much be able to predict that people with Down syndrome are going to have the pathology. What are some of the things that you're thinking about now that might modify or might be players in the brain that change, either accelerate or delay times before people develop symptoms when they have the pathology?
Right. Well, I think for me, I think that one of the most important things is the immune system and how the immune system plays a role in the brain. So we all have immune systems, and the immune system in the brain is a little bit different than in the periphery of the rest of the body from here down. There are specialized immune cells, such as microglia and astrocytes. Those are two of the several cell types that are part of the immune system in the brain. And there's also a lot of new interest and research looking at whether or not the periphery, the immune system in our bloodstream and in the rest of our body actually talks to the immune system in the brain. There's a lot of new research coming out about that. We're very interested in microglia in particular, which are immune cells that hover around the brain. They survey the brain and try to make sure everything is okay. And in Alzheimer's disease, those microglia react to plaques, they react to those tau tangles, and they start secreting proteins called pro-inflammatory molecules that might be causing some of the damage. There are many other modifiers of microglia and astrocytes, and very recently there have been a number of genetic variants that have been found in immune molecules that can actually increase the risk of Alzheimer's. They don't cause Alzheimer's by themselves, but they may increase the risk of someone getting Alzheimer's. And somebody might have one, they might have more than one. And so I think there are lots of different risk factors for Alzheimer's. And I think, as I was mentioning to you, I sort of view this as like a big river that's being fed into by different tributaries. And these different genetic variants for these immune molecules might represent different tributaries feeding into this river. And there might be other risk factors, diabetes or an ApoE4 allele feeding into this river. But eventually that river is going to hit an endpoint, a dam. I call it dam Alzheimer's. But it's going to hit an endpoint, and that's where you're going to start seeing the damage to the neurons, and then you start seeing the clinical symptoms.
It's really a complicated interplay and we focus so much of our attention often on the neurons themselves, but I think, as you're saying, the many, many other cells, including these astrocytes and microglia that make up a lot of the brain cells, are very much part of what's going on in Alzheimer's disease. I think you and many others for years have thought that. What is it that you're most excited about along the lines of what you were talking about right now in your studies of these cells and the pathways that may influence whether they help with resilience or help protect the brain against some of the problems that are accruing, or maybe contribute to the problem and make it worse?
Right. Well, one of the immune pathways that we're studying right now is called the complement cascade, the complement pathway. And so the body has this host defense system against injury and against pathogens, so against bacterial infection, for example. The body revs up this system, and it's a complicated system. It has more than 30 molecules, but it revs up this system, it activates it, and then these different molecules can instruct cells like microglia in the brain or macrophages outside of the brain to then chew up and get rid of that particular pathogen. And in aging, we know that in aging, complement goes up with aging in the brain, and in Alzheimer's disease, it goes up even further. And so we have studies in mice where we've done PET scanning with a tracer. Instead of amyloid or tau, we use a tracer that is binding to microglia and is thought to reflect inflammation in the brain. And we can see that when we compare young mice to old mice, we see an increase in the uptake in the tracer. So in your pictures, more of the red signal in the mouse when it's an older mouse. We've also seen that in an Alzheimer genetically engineered mouse model, we see an even higher uptake of that tracer into the brain and even more red. And that kind of tool is really great for studying treatments and looking at the longitudinal effects. So you can, in the same animal, you could do a pre-scan and then give your treatment and do follow-up scans at different points down the line. And it's very similar to what is now being translated into human clinical trials.
Yeah, I mean, it's really exciting that we have some of these same kinds of PET tracers that can be used in humans, and they're still in the early stages of use, but it's a real opportunity to translate from the animal work to what we can measure in humans.
Right.
So how should we think now about whether there are things outside the brain within the body? Last year we heard from Dr. Tansey about his thinking about infections and response to infections. What's your thinking about what happens to the body, maybe through the gut or through the lungs, that interacts with the outside world? Does that influence anything about what's going on with regard to the immune system within the brain?
Well, I think it's very, very important, and I think the jury is still out how much influence that has. I mean, that's a very active area of research at this time. As I mentioned, I'm very interested in the complement cascade, and complement is part of the immune reaction to pathogens, to infection, for example. And there have been studies, and there is some evidence for bacteria in the brain, viruses in the brain, and that may also contribute to this complement activation. And it's been shown that complement, for example, plays a role in the development of the brain in terms of refining the neural networks. And so in utero, complement, and after birth, complement actually works together with microglia to remove or eliminate weaker synapses. We're all born with way too many neurons. we have, as the brain is developing, we have more neurons than we need and more synapses, the connections between neurons. And complement plays a role in helping to prune back and just keep the strong neurons, the strong synapses, connections between neurons, and remove the weak ones. And what we're afraid of is that as people age and have neurodegenerative diseases in general, and in Alzheimer's, that there's activation of complement again in the brain, and that as sort of a byproduct or a bystander product, it's actually causing some removal of these synapses that may be weakened by the disease process. And it might not just be specific to Alzheimer's. It may be important for a number of different neurologic conditions and neurodegenerative diseases.
And Bob Terry was really the pioneer that talked about the importance of the synapses as the final common mechanism for cognitive impairment. So it brings your work back full circle to some of the things that I'm sure you learned with him. Yes. If you had to speculate, or maybe you have data, we heard from Dennis Selko last year too, about the thinking about pieces of amyloid that are floating around and the role of plaques as potentially this sort of dynamic equilibrium between pieces of amyloid that are floating around and maybe causing problems with the way the synapses work versus plaques that get deposited and that we can see and maybe try to remove. How do you think of of complement or of the activation of microglia or other cells as being related to that equilibrium?
Well, I think it depends first on the stage of the process. So in very early stages, if you have microglia that are competent, so microglia, it's been shown in Alzheimer's disease, are not working as efficiently as they should be. And so if you have, and there's sort of an imbalance in whether they're being protective or they're secreting all these pro-inflammatory molecules. So it's sort of the good and the bad side of microglia. And so the balance is lost during Alzheimer's disease. And so one idea is to try to restore that balance. And one way of doing that might be to block complement in the brain. You wouldn't want to block it everywhere because we need complement to fight off infections. But to be able to specifically target it within microglia or perhaps astrocytes, it's also made by them. And we need to learn a lot more about that, which cell is important. But to target that as a way to be as a way to protect synapses and neurons, even after amyloid deposition has occurred. One study that we did in mice showed that if you block the complement pathway throughout life in a mouse, the mouse is actually protected against the loss of synapses and cognition. The mice acted at 18 months of age just like a wild-type mouse, just a regular mouse. So they didn't show the cognitive impairment that the Alzheimer transgenic mice often show. And yet when we looked at the brains, they had more plaque deposition, not less. And that's because complement helps microglia chew up the plaques and get rid of it. And so to me, that means that we now have an opportunity, not just with complement, but with these new immune modulators and targets, to maybe change the immune response to these different lesions that are occurring in the brain, and maybe that will help spare the neuron loss and help promote healthy connections between neurons.
That's amazing work, and Dr. Arnold and I were just talking earlier about some of his work and some work out of Teresa Gomez Isla's lab that shows similar kinds of things in humans with some people who are cognitively normal but have plaques in the brain before they passed away were different because they had less activation of some of these immune pathway processes that you're talking about. So there are a lot of links, I think, between the studies you can do in animals and what's been observed in humans. So I was mentioning this to you when we talked earlier, but the field of cancer treatment has been revolutionized by a lot of these targeted immunologic therapies. Do you see, in talking about how you don't want to turn off complement or some of these processes globally, because it would put the body or the brain and the body at risk for lots of other problems, do you think that we'll see a point at which we can do this kind of specific targeting towards certain types of cells or towards certain components of the pathway that fine-tune it, allow us to be fine-tuned so that we're not causing more problems and we're really helping to dampen down the problematic component of the pathway? Do you think we'll get to that point?
Yes, I do. I'm very optimistic about it. I do think that we are on that road, but that a lot more work is still needed. I mean, initially with both cancer therapy and amyloid immunotherapy, we took sort of the sledgehammer approach, as we discussed, going after this big, large molecule and that's present throughout the body. And now I think there's a lot of effort, including with aducanumab, to target more pathogenic forms of amyloid, for example, and there are studies looking at tau as well. And I think that we'll be able to eventually find ways to tailor these different therapies. Complement, for example, we might be able to find a way to target a treatment that just turns off using some molecular mechanism, siRNA, something like that, to just turn off complement in the brain and not in other parts of the body or just in specific cell types. So I think that is on the future, on the horizon.
That's amazing. I hope we have time maybe in the question and answer period to talk a little bit more about some of the evolution of immunotherapy, even just within Alzheimer's, just because, as you mentioned at the beginning, it was fraught with problems that related to the fact that we didn't really understand how to move from a sledgehammer to a precision type of approach.
Right.
So thank you so much for explaining all of this to us, Dr.
My
Lemire.
pleasure. Thank you.
Thank you. Okay, so that was a wonderful start, and now we're going to move to Dr. Stephen Arnold. Dr. Arnold is the translational neurology head of the Interdisciplinary Brain Center at Massachusetts General Hospital and Harvard Medical School. Dr. Arnold has conducted research for years on neurodegenerative disease pathology and on the molecular biomarkers of these pathologies, with current interests focused on biofluid in clinical trials of aging and dementia, metabolic and vascular risk factors for dementia, and protective factors that account for cognitive resilience in normal aging. Please join me in welcoming Dr. Arnold. So we have this tradition of putting people on the spot to just ask them for a snippet of what got them into this field.
Well, I think, like a lot of us, I was, even in college, I was interested in the relationship between the brain and the mind, and that kind of continued for me. It was a fascination, and I looked at it from in philosophy and science, neuroscience. In medical school, I was trying to decide between psychiatry and neurology, and ended up going into psychiatry first, and then not feeling quite satisfied that I knew enough about the brain. So I learned a lot about the mind. So then I did neurology. And as a resident in neurology, my senior resident is our colleague, Brad Hyman, and he invited me into the lab that he was working in, and there they had this seminal finding that tangles, affected the hippocampus first and foremost in the brain of any, and for me that was like, okay, so you've got this complicated problem of dementia, and you have a very specific cells that are actually in the brain that are degenerating in a very selective way, and I found that fascinating. And then, well, why is it that those particular cells in the hippocampus are degenerating with these tangles? And one question leads to another, and then here I am, 25 years later, having done laboratory work, looking at the correlations between pathology and what the clinical profiles were of people during life, by looking at their brain tissue after life, developing new ideas about how tangles are related or plaques are related, and metabolic factors and inflammatory factors. And at some point you get a little frustrated. You've got all these ideas in the lab. How can we get them into people? How can we, and to really test out, can we do better for diagnosis? Can we do better for prediction? Can we do better for treatment and prevention? So that's been the trajectory of my interest.
Thanks. We talked earlier about how you have believed for a long time that Alzheimer's disease is not just about amyloid plaques and tau tangles. Can you tell us a little bit about some of your thinking on these other mechanisms?
Yeah, so that's the, that kind of evolved, that kind of thinking evolved in myself and a number of other people. One of the studies that I had been involved in for a long time was based at Rush University in Chicago, the Religious Orders Study and the Memory and Aging Project. And here we have about 1,200 elderly nuns, priests, and brothers who all agreed to, they were all normal when they came into the study, they all agreed to have annual cognitive testing and functional testing, and they all agreed to donate their brains after they died. And then there's another complementary study with non-clergy called the Memory and Aging Project. And when you start looking at 800 cases and you're counting the number of plaques and tangles that exist in the brain, and you're looking at it in relationship to what their memory was like 6 months before they died, some interesting things come up. Yes, there's a very strong correlation. The more amyloid plaques or the more tangles that a person has in their brain, the more likely they are and the more severe their memory impairment is. But then there were these interesting people. There were like maybe 6 or 8% of people in this group that their brain tissue was filled with amyloid plaques and tau tangles, and yet they didn't have dementia. And that's what we would now call, I think we use the term resilience. So why is it? What is it that allowed people, it wasn't a one-to-one relationship. And you get very curious as to why, what is it that allows one person to be resistant, and yet the next person with just as much Alzheimer's disease pathology in their brain to have a different profile. So it got us and a number of people thinking that, yes, amyloid and tau is how we actually define the disease, but there are other factors that are actually perhaps as important or more important at doing the damage in the brain. So inflammation is one thing that, Cynthia's our superstar for helping us understand, where that, how that contributes. Different metabolic factors, insulin resistance, and cholesterol and fats, and different vascular risk factors, oxidative damage to the brain, vascular injury, small blood vessels in the brain, and how they might become leaky, and what does that do when the brain is exposed to everything flowing through the blood system. So all of these things, and I think a number of us conceptualize these as vicious cycles. So, maybe amyloid starts something and elicits an inflammatory response, and then the inflammatory response is unmodulated, and you get a pro-inflammatory flood, and that actually causes more amyloid to form. And then the amyloid is a sticky protein, and it somehow promotes tangles, and then you get these vicious cycles going on. And so it gets really, really complicated. But the plus side of that is that there may be multiple angles that you can actually come at it. So yes, you can go after amyloid. Yes, you can go after tau, but maybe you'd get more bang for the buck if you went after inflammation or metabolism or something like that. So that's been kind of like I think a fascinating and I think really important kind of approach that now we're able to appreciate some of the complexity of Alzheimer's in a way that we haven't before. And it's presenting a lot of new opportunities for intervention.
When people sometimes talk about metabolic issues in Alzheimer's disease, sometimes they refer to it as type 3 diabetes. When we talk about metabolic abnormalities with things like insulin degradation and processing and that type of thing, do you think of it as something that we really should not think of as like diabetes? Or do you think that it's related enough that we should conceptualize it that way, partly maybe because it gives us opportunities to think about some of the things that we're going to talk about with Dr. Budson about possible lifestyle decisions that people can make to do something to reduce that risk?
Right. So that's a great and really fascinating kind of, and I'm using the word complex a lot tonight, but very, very complex thing. But what I think some of our work has shown, as well as others, is that, so we actually got interested in this because people with diabetes, and in particular type 2 diabetes, the adult onset type of diabetes, that where the body is resistant to the effects of the insulin, and so your sugar goes up. And sugar is the key, that's what diabetes actually means. The brain, insulin is an important chemical in the brain too, but it doesn't really affect sugar so much, glucose. It acts more like a growth factor to sort of promote the healthy, to sustain the brain cells and promote the growth of brain connections and the maintenance of brain connections. And so insulin, but what we started to do is looking at brain tissue in people who died with Alzheimer's disease. We found that there was a lot of evidence that brain cells were resistant to insulin. And so there's an analogy between the type 2 diabetes in the body, which causes increased glucose, but perhaps insulin resistance in brain cells that may have some other effects, but just as powerful, not related to sugar, but more related to the health of brain connections. And so this observation, I think, also is really important because we've got a slew of medicines that have been developed for diabetes, for type 2 diabetes. What if some of those medicines that help insulin sensitivity in the body, what if they also make brain cells sensitive? So that opens up. Now we can actually use some of these observations and repurpose a drug that was developed for the diabetes in the body that no one thought would have anything to do with Alzheimer's disease, and repurpose that drug, and let's see if it actually helps Alzheimer's.
That's fascinating, and it definitely shortens the time it takes to figure out how to give it to people, if it's going to be safe, what doses to give, maybe. We probably don't have time to talk about the metformin work you've done, but maybe we can come back to that in the Q&A period. Could you just say a few words about this idea of translation and the importance of biomarkers? I think Cynthia touched on it, but you do a lot of that work, and so how can we measure some of these kinds of things in people in ways that we could potentially link to measurements that we can make in animals so that we can close that loop more efficiently?
Yeah, so we're talking about translation at multiple levels, between animals and humans, but also between humans and neuropathology in the brain. So, still we tell people that the only way you can be 100% sure is if you have Alzheimer's disease is if you look at someone's brain tissue under a microscope and look at the tangles and plaques. And that's I think still true, but the huge development in the last 10 to 15 years has been this biomarkers. So we now have amyloid imaging. We now have tau imaging. We can measure amyloid and tau and much, much more in spinal fluid, inflammatory markers, metabolic markers, all these things that we've been talking about. And the other thing, because you can measure those things in humans and they strongly relate to what's actually going on inside the brain, those are measurable things in animals as well. So you can look at the same kind of mechanisms in our animal models, in our human people, and relate that to what has always been, since 1900, 1906, when Alzheimer first looked at a person's brain under the microscope, what he described. That really defines the disease. Biomarkers are transforming the field, and also for clinical trials as well. Before we had these kind of biomarkers, even experts, who see Alzheimer's all the time, would be wrong with their diagnosis 20 to 30% of the time. And now, with some of these biomarkers, we have then, we're correct maybe 95, 98% of the time with very few mishaps.
And may be able to measure some things that we haven't even thought about measuring in the traditional pathology sense.
Right.
Well, thank you so much, Dr. Arnold, for going through all of that with us. I know it's the tip of the iceberg, and hopefully we'll get more discussion. So keep track of your questions. Our team is circulating with pieces of paper to write questions down on, so I know it's a lot of material, and feel free to capture some of it now and hand it in, and we'll have a lively panel discussion shortly. Our third speaker tonight is Dr. Andrew Budson, who's a professor of neurology at Boston University, a lecturer in neurology at Harvard Medical School, and chief of cognitive and behavioral neurology at the Veterans Affairs Boston Healthcare System. His current focus is on educating healthcare professionals and the public on better understanding memory disorders, the importance of early diagnosis and treatment, and how diet, exercise, and other strategies can potentially help. Please join me in welcoming Dr. Budson tonight.
Okay. Well, thank you. It's a pleasure to be here.
So give us your backstory about how you got into this field.
Absolutely. So I think perhaps similar to Dr. Arnold, I was interested in the brain and the mind and how these things went together. When I was an undergraduate, I actually double majored in chemistry and philosophy to try and sort of get at it from both sides. But what really got me interested in Alzheimer's in particular, and the research that I do, is I was taking care of a whole bunch of different patients during my fellowship in memory disorders, and some of these patients would have memory problems due to strokes, other of them had memory problems due to tumors, one had memory problems due to encephalitis, and we were able to work with our cognitive occupational therapist to set these individuals up with pill boxes, calendars, reminder systems, and they could basically live independently and go on about their lives. I then took care of a number of patients with Alzheimer's, and I tried to set them up with the same types of systems and calendars and notebooks, and it didn't work. It would not work to have them, continue navigating through life. And what we began to realize is in Alzheimer's, not only is there forgetting, but there's also false memories. There's also thinking that one took that medicine or turned off the stove when they didn't do those activities. And because of that, they would look at the pill box and they'd say, oh, I already took my medicine. I don't need to lift up the little cover. And you'd see a sign above the bed that says, go check the stove. And they'd say, oh, I don't need to check the stove. I remember, I turned it off. And that got me interested in looking into false memories and working on what are simple strategies that people can use to both improve their memory for true information and also reduce their memory for false information.
It's really a fascinating insight and something that I think we all need to learn how to make use of. You touched on how there are a variety of contributors to problems with memory. Could you give us your Memory 101
Sure.
discussion about how memory really works?
Yeah, absolutely. So the way I think about the memory system, I think about a filing system analogy. And there are two important components to this system. And the first is our file clerk. So our file clerk is actually our frontal lobes right behind our forehead. And it's the file clerk's job to take information in from the outside world and put it into the physical file cabinet. When one needs to retrieve the memory, you can picture the file clerk pulling open the file drawer, looking through the different files and pulling out the memory that you're trying to retrieve. Now that's all good, but as we're all getting older, what happens? Well, our frontal lobe file clerk is getting older too. And in an analogous sort of sense, our frontal lobe file clerk is getting a little hard of hearing. What? And because of that, information may need to be repeated a couple of times in order for the file clerk to get a hold of it. And that's just part of normal aging. Now, the next thing that happens is our frontal lobe file clerk doesn't move quite as quickly as he used to. And because of that, in part of normal aging, it may take a little bit longer for us to have our frontal lobe file clerk move over to the cabinet and pull out that information that were needed. And lastly, our frontal lobe file clerk just doesn't see quite as well as he used to. And so as he's pulling open the file drawer and looking through the files, he may need a hint or a cue in order to retrieve the right information. And all of those are part of normal aging. Now, in Alzheimer's, what happens is the file cabinet gets affected. Now, the file cabinet is another part of the brain. It's the hippocampus that we've heard about today. That's the place that Alzheimer's disease typically attacks first, that and related structures deep in the temporal lobes. The temporal lobes are just behind your eyes, by your temples. And the way I think about it, what Alzheimer's does is imagine if you pull open that file drawer and you look down inside and there's a big hole in there, okay? So you could have the best frontal lobe file clerk in the world taking information in from the outside world, putting it into the file cabinet. But what happens? It goes down through the hole, gets lost, never to be retrieved again. And so with Alzheimer's, Even if information is repeated, even if we wait a little bit of time, even if we give a hint or a cue, we cannot get those memories out. And when that happens, we call it rapid forgetting. And that's never normal and should always be evaluated.
Thank you. That's a wonderful analogy. I love how you continue to develop it over time. It's
continuous improvement.
Yeah, it's great.
It's better with age, like a fine wine.
It's great. I mean, I think we talk a lot in these sessions about how challenging it can be to differentiate between early stage Alzheimer's disease and what may be a different illness or what might be normal aging. So I really like the way you unpack that. How do your thoughts get provoked in hearing our prior two speakers talk about some of the processes that may be more modifiable by some of the decisions that we make about how to lead our lives in some of the work that you've been doing, and you take care of patients with these diseases and their family members ask you about what they can do. You try to help them. You've written a book to try to help give people specific guidance about some of these topics. Help us understand more about how you might be thinking, especially in the context of what we've just heard tonight.
Sure. There's sort of three or four different ways that I think about what both individuals that are showing signs of early memory loss due to Alzheimer's disease, but also what healthy older adults can do to try and keep themselves and their memories as strong as possible and reduce their risk of developing Alzheimer's disease, or perhaps delay the onset of it. There's a number of studies, some of them came out just recently, that suggested that people that started in middle age to eat better and exercise more were able to delay Alzheimer's disease, if it was going to come, by up to 5 years, which is very exciting. So specifically, let's start with the different types of things that people can do to strengthen their memories and keep their memories strong. So it turns out that if one spends time doing crossword puzzles or Sudoku or brain training games, most of the evidence shows you get better at crossword puzzles and Sudoku and brain training games. There's not a lot of evidence that it really translates into overall brain function. Although lots of different laboratories, including several in the cities, including our own, are looking to see, can we find some type of cognitive stimulation that really will provide that? But in my opinion, we're not quite there yet. So what can you do? Well, the first thing that I would say in this type of brain strengthening strategy is social activities. Social activities are so important, and the opposite can be very damaging. So unfortunately, sometimes when people develop memory problems in Alzheimer's, they end up becoming isolated and not being able to be into all the different things that they have done before. And that can make cognition worse. The next thing that's been shown to be important is doing something new. So if you're going to do something with your brain, learn something new. Learn yoga, learn pottery. Those are the types of things that have been shown to be important. If you're traveling for a trip, learn about the new place you're going to go. And the last thing is having a positive mental attitude has also been shown to be very important, and this may be partly because if you have a positive attitude about aging and life in general, it's going to help you to eat well, exercise more, take care of yourself, be social, those types of things. So let's talk about eating well. So there are many different studies that are out there that have looked at different diets. And if you sort of group them all together, in my view, what they all have in common are some of the elements of the Mediterranean diet. That's the thing that study after study, both epidemiology studies as well as actual clinical trials, have been shown as beneficial. So that includes eating things like fish, olive oil, avocados, fruits and vegetables, nuts and beans, and whole grains. So those are the things that have been shown beneficial. So what are the things not to eat? Well, it's sort of everything else. So, we were just hearing from Dr. Arnold talking about how sugar may be bad for the brain, and insulin resistance is something that may be happening. So as far as we can tell, all of the studies suggest that having a lot of refined sugar, a lot of white flour, white bread, normal pasta, which all get broken down into sugars very quickly, is really probably not good for the brain. Try and stay away from that. That's more once in a while type food. Same thing with red meats, fatty foods, things like that. And the last, but not the least, probably the most important thing is exercise. In fact, we've all been sitting for a while. Would it be okay if everybody just stood up for a minute? Let's all just stand up for a minute. Let's have a stretch, okay? Move your feet a little. Don't kick your neighbor. Move your arms a little. All right, good. Okay, so that's very important. So exercise does so many amazing things for you. I'm sure I don't need to tell you that exercise can help prevent heart disease and strokes. Did you know exercise can also prevent falls, okay, which can damage the brain? Exercise improves mood. In fact, it improves mood as much or more than many antidepressants that are on the market today. Exercise improves sleep, and we know that sleep is important for being able to pay attention because you're not feeling tired. We know sleep is important for memories going from a short-term temporary store to a long-term permanent store. And there's even studies to show that the clearance of beta amyloid happens when you sleep. So sleep is critically important for memory. But the most exciting thing I think about exercise is that exercise can actually increase growth factors in the brain that can promote the growth of new connections between neurons and perhaps new neuronal development themselves. And the increase in these connections and perhaps neurons is so great that you can actually see it on an MRI scan. There was a study that came out in 2011 that showed that those people that engaged in aerobic exercise showed an increase in their hippocampus, in our memory file cabinet, in as short a time as 6 months. And that the amount of change that they saw in the hippocampal size correlated with their memory performance. So people ask me, Dr. Budson, is there a magic pill to help my memory? The answer is yes, there is. It's called exercise.
Thank you. And how many miles did you ride in Vietnam recently on your trip?
We rode a lot of miles in Vietnam and saw a wonderful country.
I can't say that Dr. Budson practices what he preaches. I'm just trying to find more excuses to avoid getting pulled into the Sunday morning bike rides with Jim Wessler, I think.
There you go.
So it's fascinating work. Unfortunately, we have to move to the panel discussion, which I'm sure will give us plenty more opportunities to talk about this. And you can just stay up here. And thank you very much for that insight. So, come on up, guys. Sure. Start passing your questions around. We're going to get everyone situated up here. I don't know if we need the table. Well, you could put your water on it, probably. Wherever you want to sit, maybe girls in the middle. Thank you. Okay, so thanks for writing everything down here. I think the first question is a very interesting one, which is what do we think about, is it helpful for patients with MCI or dementia to know the status of amyloid in their brain? So I'll leave that one to Dr. Arnold, maybe.
Oh, boy. I think each of us is our own expert in that regard. For right now, I think that there are, it's a personal decision, and it's a personal decision until we actually have a definitive way to prevent progression moving on. And I think this is actually getting even more important as we move even earlier than MCI into what's called preclinical Alzheimer's disease. So I think one of the things that we've learned from all these biomarkers is that Alzheimer's disease pathology process probably starts 10 to maybe even 20 years before someone notices that they have even a little forgetfulness. And yet we don't, I think that knowing that at that stage, and probably in the MCI stage, does give us the opportunity to, and perhaps the motivation to change the lifestyle that we have, to start exercising, to pay more attention to our diet, to pay more attention to our activities, and that's really important. So yes, that is I think the advantage of knowing there. It will become, if we do end up with a successful treatment or prevention, then sometimes I use these biomarkers as, well, you turn 50, 55, you get your stress test, you get your colonoscopy, and you get your amyloid PET or your spinal tap, and if any of those are positive, you do something about it, and that's what we're really moving towards.
Thank you. There's a question here that I think you each mentioned something related to, and maybe if Cynthia might want to start with this one. The question is related to is there a relationship between CSF flushing of the brain, cerebrospinal fluid flushing of the brain in sleep, and the microglial or inflammatory processes that we talked about in relation to amyloid or potentially other mechanisms that are contributing to the problem?
Right. Well, that is a very interesting question, very sophisticated question.
I warned you about the audience here.
I know. I think there is very clear-cut evidence that there is a clearing of beta amyloid, especially during sleep. How and whether microglia are involved in that is still not crystal clear yet. It would make sense that glial cells of some sort or immune cells in the brain of some sort may be involved, but there are these little teeny tiny cells called pericytes that line the blood vessel walls in the brain. And I think that those are probably also critical players in the removal of amyloid, in the normal removal, because keep in mind, beta amyloid crosses the blood-brain barrier in both directions. So it's not that it's only in the brain, it's in the blood as well. And so it's going back and forth. But I think the clearing part of it may actually be in part mediated by microglia, and then perhaps by these pericyte cells. And it's also important to keep in mind that these astrocytes, the other glial cells, line and form a barrier around blood vessels in the brain. And so I think there's a very good possibility that these different immune cells are playing a role in that clearance process.
Any other comments on that? Okay.
No, I mean, I think it emphasizes the importance of sleep as being very important for the health and kind of prevention of any progression or development of Alzheimer's.
I'm happy I didn't see anyone taking that to heart tonight. And this is a question that I think both Andrew and Stephen could address, and I'm sure Cynthia would have interesting things to say about it as well. My mother was diagnosed with early onset Alzheimer's disease at age 60, and a genetic component, the apolipoprotein E allele, was identified. What can I do as a 30-something-year-old to get ahead of this disease, whether it's through lifestyle or possibly participating in studies?
Well, I'll just mention the lifestyle things that we just talked about, although the things I mentioned were specifically things that older adults and people with early stage Alzheimer's disease can absolutely benefit from, but 30-year-olds can absolutely benefit from all these activities as well. And there's increasing evidence that it is, although any amount of exercise, diet, and socializing is helping, it is probably even more important to start early. So if you're a 30-something and you have a family history of this, you really want to work on these lifestyle changes. And I'll let Steve talk about some of the clinical studies.
Well, no, I mean, I think I'd emphasize, I just kind of back up what you say, that the window of opportunity is greatest the younger you are in terms of developing these habits. You know, changing your lifestyle is not easy. Sometimes I think it's easier to change your genes than it is your lifestyle. It's hard. But the earlier you are able to develop those habits, the more it's going to pay off. And you know, for a 30-year-old, that's really quite young. You know, there are clinical trials now, the A4 trial, the A5 trial, that are targeting people who have signs of amyloid in their brain by biomarkers, but they don't have any symptoms. And if you, from a clinical trial standpoint, this is the important and critical experiment that we do, if you are able to lower amyloid levels in the brain, can you prevent the emergence of mild cognitive impairment in years? So it's really important that we you know, those of us who do have some increased risk, that we're able to participate in such trials.
And along those lines, there are several questions, partly foreshadowed by some of the things that were mentioned tonight about the A4 trial and about the Pointer study. Would any of you comment a little bit more on what those are, and you know, maybe there are people through the trial match system or other mechanisms that might want to consider enrolling in one of those trials?
I can talk a little bit about Pointer.
Yeah.
So Pointer, and I'm not a clinician, keep that in mind, that's my disclaimer. But Pointer is a large study that's just getting underway now, sponsored by the Alzheimer's Association. And as Jim mentioned earlier, it is going to enroll 2,500 people between age 60 and 79. And these are people that do not show signs of dementia at all, but who are not necessarily have not necessarily changed their lifestyle habits. So they're people that tend to be more sedentary, maybe overweight, not getting great sleep, and not having the most healthy diet, not exercising on a regular basis. And so the idea is to take this at-risk population of people while they're still 60 to 79 years of age, and trying to help them by a real hands-on advice and guidance with their diet. So it's the Mediterranean DASH diet. So again, the same type of diet that Dr. Budson was describing. Exercising at the YMCA 4 times a week. And so it's not just going in and doing random things at YMCA. It'll be an actual structured program, and people will work with trainers, and it includes aerobic exercise as well as resistance training, weightlifting, that kind of thing. And it will be tailored to people so that, you know, you won't just jump in and start doing all of these exercises at once. People will ramp up, starting slowly and ramp up over time. This is a 2-year study. So the first, I think, 4 months is dedicated to sort of ramping up and getting used to this. Another component is social and cognitive stimulation. So there'll be cognitive programs on computers, cognitive tests and stimulating activities that people can do on computers. But there's also engagement once a month. The group will get together, the local group will get together for social activities and cognitive stimulating activities. I'm trying to remember. Is there one more? I think that's sleep is not actually.
So can I just ask, what happens in the control group? Is there a control group?
Yeah, there is a control group, and so the control group.
Do you have to sit on the couch and watch TV?
Nope. Nope. So the control group, instead of this very intensive meeting, you know, with basically like guidance counselors for health, on a regular basis and going to the YMCA 4 times a week and having the social interaction every month and all of that. The control group instead will meet once a year, and they will receive guidance through the Alzheimer's Association website and other websites to learn more about, it's an education program basically, and it will be upon them to take the initiative to go out and do these different things, like, you know, to improve their lifestyle and improve their health. So it's a much less user-intensive program.
Got it.
Thanks for those details. I mean, it definitely sounds like a challenging study to design and to carry out, and to be confident that you're going to really have a group of people that are engaged enough to be part of the study, but aren't going to do very much as your control group. I mean, I think it sort of stacks the deck against us a little bit in finding effects. So, but as you say, it's really the first study of this magnitude addressing these issues in a prospective way. So I think we'll learn a lot from it, even if some of what we learn is how to do it better next time. Hopefully it'll work, but a lot of times we learn each study we do how to do it better next time. So Stephen or Andrew, do you want to just mention briefly the A4 trial and what Risa and her team are trying to do for people that may not be aware?
Just, very briefly, this is a prevention trial. We think that people who have amyloid that's developed in their brain are at high risk for progressing to mild cognitive impairment and subsequently Alzheimer's disease. There are anti-amyloid drugs. The idea is that if you can lower amyloid levels in the brain, perhaps you are starving, that you are taking away the fuel or the energy that is the pathology really that is going to progress to the point of developing symptoms. And so it's a big commitment for people to come in, to get infusions, an intravenous infusion of an immune therapy. I think it's once a month. I can't remember. But it's really it's going to be a really critical study for us to learn whether we can actually prevent Alzheimer's disease by reducing amyloid in the brain.
And I'll just add that some of you may have heard about on the news that some of the anti-amyloid drug trials have failed. And so a lot of us are sort of wondering, well, what is the reason? Why have they failed? And one hypothesis is that, well, the problem is we didn't start early enough to get that amyloid out of the brain before it caused inflammation, before it caused tangles to form, other metabolic disturbances. And so this is a trial that is going to be testing that early hypothesis, well, hopefully helping, thousands of people.
And it may be, I mean, a lot of people are investing in the idea that just because a drug, as you say, Andrew, doesn't work after the degeneration has already caused symptoms of dementia, doesn't mean it might not work if you give it to someone before the degenerative process has gotten underway.
Exactly.
Right. So it's a bet, it's a gamble, but I think we're going to get an answer. I think the studies and other studies like it are designed in a rigorous way so that we'll get an answer. And be able to learn, hopefully, from what we hope will be the beginnings of the success in this arena. So it's really revolutionary in many ways that we're doing both of these types of studies and targeting efforts to modify the disease, both through drug treatments and through lifestyle-related interventions, and really mobilizing a lot of people that it takes to be engaged in this kind of research in order to get these answers. So it's really a testament to people's persistence and commitment to be in these studies. So a question for Dr. Lemire is about, this always comes up in this part of the country, Lyme disease. So do you think that there is any evidence that there's a connection between exposure to Lyme disease-related pathogens and processes that might contribute to the Alzheimer's disease or to some of the inflammatory systems that might make people more vulnerable to disease when they have plaques?
Yeah, that's a very interesting question. I wish I had an answer for that, but I don't. I would say that anytime you have a situation where you have a strong inflammatory response, it is possible that that is going to affect the brain in some way. And as far as I know, I don't know myself of any studies that have found direct correlates between Lyme disease and Alzheimer's disease, any link there. But I do still think that there is a potential for inflammation in general, whether it's through Lyme disease or some other inflammatory process going on long term, for example, chronic arthritis, something like that, that may actually set up the brain so that it's now more vulnerable. And again, it wouldn't necessarily cause the disease, but it might make it more vulnerable, make the person more vulnerable.
I think we get asked as clinicians all the time this question because Lyme disease exposure is so endemic to this part of the country that people will get a positive Lyme test and want to do something about it because maybe we could do something about at least part of what's causing this person's memory loss. So, Andrew, do you have thoughts about how people should
Right.
think about that?
Sure. All I was going to say is that I do think if somebody has had an exposure to Lyme, maybe you had a rash, or you don't ever remember a rash, but you take lots of walks in the woods and you pick ticks off yourself, or your spouse does. It's worth getting checked out. I don't know, just like you say, that it necessarily causes Alzheimer's disease, but it is a rare but potentially treatable cause of cognitive impairment. So I do think if you're somebody that has active Lyme disease, that it's worth getting treated, getting evaluated from your regular doctor.
Yeah, I think the thing that I would say too is that the flavor of the symptoms that people have, at least that I'm aware of, that have Lyme-related encephalitis is quite different than what you see in a person with most forms of Alzheimer's disease or related disorders. And I think some of this comes back to the question of what biomarkers we have to support our diagnosis in a person to begin with. And if we have evidence that the person has the kind of problem in their brain that goes along with the kind of problem in their memory or other cognitive systems that we associate with Alzheimer's disease, I think it's a pretty big leap to imagine that Lyme could be playing much of a role in that, even though, of course, we don't want to miss something that could be making it worse than it would be. It's a tough situation to think about. So, question for Dr. Arnold. Can you say a little bit more about the metformin work that you've done?
Okay, so this relates to when I was talking about insulin resistance, and so we decided to run a small clinical trial just to kind of probe the brain to see what the effects of metformin, which is the most commonly prescribed medicine for type 2 diabetes in the world. I mean, millions of people are on metformin. And so we recruited some people with mild cognitive impairment or early Alzheimer's disease who did not have diabetes. So they were non-diabetic, they did not have any need, and we wanted to see what would metformin do to the brain. It was a short study, it was a pilot study. We didn't see any major changes in terms of memory. It's not like the light bulbs went back on. We did see some improvement in cerebral blood flow, so it looked like some areas of the brain, particularly in the frontal lobes, were more metabolically active with that. We are still looking at some of the spinal fluid tests that we did in those people to look to see which inflammatory markers or metabolic markers may have moved in a healthy or unhealthy direction. Hopefully healthy, but we're still examining that. But I think it was really, we used it as kind of like a probe. Could we do this type of study? Could we see any signal in the brain that would prompt us to do a more ambitious, larger trial with metformin or another drug? And I think that some of the positive findings that have emerged from our analyses so far suggest that it is a line of investigation that's worth pursuing.
Thank you. Just maybe a couple more questions, I think. So how close are we to having a blood test that we can do to diagnose or help diagnose or monitor Alzheimer's? You know, Dennis Selko last year said that he thinks we're not that far off from that. Maybe Cynthia or Stephen?
Okay, that's one of my pet areas. So, you know, we do PET scans are involved, they're expensive. Spinal fluid, people don't like the idea, even though I think it really has a bad rep. It's not as bad a test as people think. It's certainly a lot better than the colonoscopy that I mentioned before. It's really quite a trivial procedure. You know, I personally do about 3 or 4 of them a week now with folks that are in our research or clinically. And we learn a lot from spinal fluid. Blood has always been the holy grail, okay? Can we get a blood test? And one of the problems that's existed is that we've just not had measures, biochemical measures, lab measures that are sensitive enough. But that's changing now, and I think we're really optimistic. There's a company in Lexington that has developed ultra-sensitive assays that we are able to measure tau in blood. We can measure another protein called neurofilament light, which is a brain-specific protein, and we can now measure that. We're not quite sure yet how well these tests are going to perform. You know, how good are they going to be at diagnosis? How well can they track progression? How good are they at staging disease? But you know, the fact that we now have these tools, I think there's potentially we're at the beginning of a new era, and it is because of some of the really extraordinary technological advances that we now have.
I'd just like a follow-up question to either one of you. I read just today that I think it was a study out of Canada that suggested there's a saliva test for amyloid. What do you think of that?
Saliva is interesting, and actually in the Quanterix system that I think we're using at Brigham and at Mass General now, that's the company here in Lexington that's developed this assay. I don't know if anyone's spit in one of these tubes, but I'm going to, and we're going to measure to see what's in saliva. They're talked about. Because there actually are some interesting, perhaps a little bit more direct connections between the spinal fluid that sort of bathes the brain and the salivary glands. It's kind of interesting.
I think, thanks, Stephen. Last question maybe, whether it's in animals or people with Down syndrome or people with sporadic Alzheimer's disease, is there evidence that trying to get people to adhere to some of the lifestyle changes that we're talking about after they already have established cognitive impairment and neurodegeneration, is there evidence that that might help them, or is this still pretty speculative at this point for people in the mild cognitive impairment or dementia stage?
I'll just briefly say that I have reviewed this, and there is definitely evidence for exercise that can help to keep people stable longer or lessen the slope of decline when MCI and mild Alzheimer's disease dementia. So I feel confident about recommending it when people are in at least those mild stages.
And I'm, again, not a clinician, but I believe, so I have a relative with a form of dementia, and I'm her caregiver, partly. And I would say that also, from personal experience, the social activity part of this is key, and it makes a huge difference. From living alone and being alone in her own place to being in a wonderful assisted care living situation where there's all kinds of activities every day, every evening. I really think that also makes a huge difference.
Oh, absolutely. And there's definitely studies to support that as well.
Yeah.
Absolutely.
Yeah. And I would agree with that. I in my own clinical practice, and my reading of the literature. Physical exercise makes a big difference. You know, when I talk about socialization or social engagement, I tell my patients, you know, there's actually nothing more stimulating to the brain than interacting with another person, reading their emotions, listening to their words, you know, being active, the emotional buoyancy that you get by social engagement. It pays dividends in so many ways, and you know, the tendency to get isolated, either because you know people are more apathetic or it's harder to do things, but you know, also the other health issues. Hip hurts, back hurts. The tendency to stay at home is strong, but it must be overcome because social engagement is really quite a powerful
Force.
Yeah, force.
Well, I'd just like to give special thanks to our speakers tonight. Thank you so much for coming and sharing all of your insights. And thanks to you for coming. Each of you received the trial match card when you came in, and as we've talked about before, we can't do this kind of work without participation, and so if you have time, please fill out the trial match card before you leave. And we're happy to stick around afterwards for a little bit and talk to people if they have other questions. So thank you so much. And by the way, the session tonight was recorded, and there will be a web stream with this URL. So if there are people that you know who weren't able to attend but would like to watch it, they're welcome to do that. So thank you, everyone. Good night.